Hi, I'm Belle. I study archaeology at the University of Southampton, and subsequently, I know nothing about biochemistry. My wonderful partner, however, is in her master's year of her biochemistry degree at Southampton, so she knows at least a little bit more than me. And so I'm going to grill her about the subject and her research to try and understand it a little bit more. Hello. Would you like to introduce yourself?
Hi, I am Anastasia. I am an integrated master's student in the University of Southampton and I study biochemistry.
Wonderful. So I guess we should start with the basics. What is biochemistry? Can you define it for me? Because there's so many different biology degrees, biomed, stroke biology. Like, what is biochemistry?
Of course, yeah. Biochemistry is also known as molecular or cellular science, is the study of everything from the DNA, but also tissues, cells, and even cancer research is included.
So you go from big to small, and then you do a bit of everything as well.
Yeah, it covers literally everything in between.
You can look at normal cells, infected cells, the communication between cells, all of that fun stuff.
And you can gain more expertise as you head through the years, and you can become an expert in one particular field.
So that said, what do you focus on? What area do you enjoy the most?
I would say before I started university that I was really interested in genetics, and when I would graduate I would become a geneticist and I would study only the genes. So that is the smallest of the smallest, the molecules that make up your genetic code, and that code makes your proteins in your cell. So I didn't think when I started studying that I would be interested in what I'm currently researching, which is cellular signaling, the communication between those cells in order for them to function.
So how does that work exactly? How do they communicate in the most simple of terms?
Well, in general, there's quite a few ways. Cells have messengers which help them communicate different things. So they can send letters or text messages between them to say that conditions are not good now to grow and they should not make any more of the cells. But also when you have hormones and neurotransmitters, we all know serotonin or dopamine. Those neurotransmitters also communicate to your cells, and when they're released, it tells the cells that, they should start producing something. So cellular signaling is basically the study of all those messages and how those messages make cells work and function, but also that then goes into how can that go wrong? Well, where does it go wrong? And can it lead to cancer? Or why does it lead to cancer?
And what led you to become interested in cell signaling?
Well, it's a funny story actually, because when I was introduced to cell signaling, I actually hated it. I did not think that genetic, what I was really, really passionate about, would tie into that much. And it also seemed like a little bit of a chore to remember all those different proteins and all the messengers, how all of them connect. It was a lot of work, because there are so many cell signaling pathways. That is the routes that the messengers can take in the cell to deliver a specific signal. When I was first introduced to it, I thought, I would never go into it. And funnily enough, when I was given the choice on my third year laboratory project, I put as my first choice genetics and immunology and pharmacology and all those cool fields. I was actually given a project about cell signaling, and I thought, no, I'm probably not going to enjoy it. It's not genetics at all. I was pleasantly surprised, because when I actually joined in the lab, it was completely the opposite of what I had thought. And actually, genetics and cell signaling merge together very well. And genetics actually can go into any field. It is useful for any other study.
So that gives us a wonderful segue into your project from last year and your fourth year project that you're finishing up this year. So what have your projects been on? Obviously it's on cell signaling, but what cells, what are they signaling? Give me the details.
Well, last year the project was completely different from what it is this year. So on my third year project, I was given a taster into what cell signaling research is like. So what I got to do, which was very, very exciting, was use CRISPR.
Oh, bringing up CRISPR, I think obviously everyone's heard of it, but can you actually tell us exactly what it is and what it does? It's to do with genomes, right?
Yeah, so in short, CRISPR is a tool that can help us edit our genetic code. So probably you've heard about it in the news. It is a promising tool that can help us fix or correct genes, especially in diseases which have some defects, and therefore that person does not produce protein. But in the laboratory, we use it so that we can alter the genetic code, or even we can delete a gene and its product from a cell. And then we compare that to the functioning cell. And therefore, if there's any differences, if the cell even functions, sometimes they can even die after deleting a gene, then we can dig deeper into, what is going on there? What processes are affected by what we've done?
So it's like when you give rats cocaine, but on like a molecular level.
Yeah, it is a way to target a specific gene. And therefore, with that, we can make a disease model and understand that disease even better.
That's interesting. So talking about your research project, what other things have you done in the lab? What kind of skills and techniques have you used?
Last year, I was able to edit the genetic code using CRISPR, specifically for a gene phospholipase C beta 1 or PLC beta 1.
Right over my head.
Which is able to break down a lipid in the body.
Lipid is a fat? Or a fatty component?
Yeah, you can think about that. It isn't actually a protein. It is a lipid that is found in our cell membranes. So it is the middleman between messages coming in outside the cell and signals going to the cell.
And then you edited these lipids with CRISPR.
Unfortunately, you cannot edit the lipids because they are not made by genes.
That makes sense. Yeah. I realised that after I said it.
So unfortunately, you cannot manipulate them very easily like we manipulate proteins. So the only way you can study them is to manipulate the proteins that change it, either that break it down or that make it. The gene that I edited, PLC beta 1, breaks it down to make two new messengers that do different things in the cell. And then I was looking into, does this cause the cell to grow or to heal wounds slower? And to do this, I did something quite cool. I scratched the cells and looked them under the microscope for a few days, measure and saw how fast they're growing, if they are affected by what I did or not.
Oh, that's interesting. And then what about your fourth year project? How different is it?
Completely different. So what I was doing then in the third year was kind of more general, because we could not see specifically those lipids, how they were affected. We were assuming that by deleting that gene, that therefore it was not breaking down into those two new messengers. But this year, I'm trying to develop a way to track phospholipids in the nucleus and see what it does in the nucleus, because currently a lot of conflicting information about it exists, that in the nucleus, phospholipids can do anything from transcription regulation, apoptosis.
Apoptosis?
Apoptosis is basically the fancy word for cell death.
Yeah, and then transcription?
Transcription is the process which copies the genetic code, the instructions to make a protein, into a new molecule called messenger RNA. Then, if you think about messenger RNA being in one language and the code necessary to make a protein being a different language,
Google Translate.
It Google translates DNA to make a protein.
That makes sense.
Unfortunately, lipid signaling research currently relies on ways we can see what the phospholipids are doing in effectively dead cells, and therefore it is not the best way because you are not able to see what it actually does.
We're working on making a way to track it so you can actually see what it's doing.
Yeah, basically we are trying to develop something that will track it to see where it actually goes in the messaging chain of cell signaling, especially in the nucleus, which is very, very important for cancer research and cancer treatment.
I see. So in the future, where could your research fit in the bigger picture?
Well, in and of itself, I'm just trying to develop a tool to be able to improve research and findings about lipid signaling in the nucleus. So hopefully it is going to be used by all of the labs worldwide that...
That's amazing.
...study these like beautiful lipid molecules that are not very well known. There's not much of them, but they do a lot. and unfortunately we don't understand them very well. And therefore studying how can their signaling, how can the messaging system go wrong in terms of cancer in drug treatment as well, because then you can see what is different between cancer cells and normal cells by just tracking the phospholipids, and therefore you can develop a drug treatment that only treats those cancer cells because their messaging is different.
Ah, I see. Okay, so thank you for going into some more detail about your projects. It's nice to know, or have an idea at least, of what you're doing and what you're studying. I thought we could finish up with just some more general questions about science and about your opinions on things. So I thought we'd start with, what are your favourite and least favourite things about lab work?
I would say my favorite things about lab work is learning all the new techniques and the different experiments that you can do so you study what you want to study. For example, one that I learned this year was cloning. So
Oh, that's cool.
yeah, that was really, really cool.
And then your least favourite thing?
My least favorite thing, I guess, is, I think any scientist or any student will relate a lot, is just experiments failing and you having to try and kind of troubleshoot. But thankfully, I will turn it into a positive. With experiments failing, you actually understand something even more, and then the next time that you do it, you learn more.
Next up, your favourite science fact.
There are so many weird named proteins. As a scientist, you can name it whatever you want. So I think the wackiest one is that there's a protein called Sonic the Hedgehog.
Oh, it's brilliant.
Other ones are Nemo. There's also one called Spock.
Wonderful. It's been really nice to talk to you. Thank you for answering my plethora of questions. That is nowhere near all of them, but it is all we have time for. So I hope you've enjoyed your time, and I hope everyone who's listening has enjoyed learning about biochemistry.
Yeah, thank you very much.